Colince Campbell, who is a postdoctoral fellow in my laboratory here. Colince hails from Cameroon, where he had a rich and deep history in mosquito biology, primarily obviously with Anopheles, and during which time he did some pretty neat work with George Demopoulos whilst Colince was in Cameroon, looking at transcriptional profiling of the M&S strains of Gambi. He then moved to Montpellier and continued on his mosquito work, and then and collaborated with Nora Pazanski and Matt Hahn at Notre Dame, and then moved back to Cameroon, and then was recruited by Brad White, who was an assistant professor here at Riverside, to start working on some interesting projects in Anopheles Gambi, primarily with recombination mapping. So without further ado, here's Colince. Thanks for talking today.
Thank you, Peter, for this introduction. Thank you for inviting me. So I'm going to present some of my recent data on the adaptation of Anopheles mosquitoes to human habitats and vector control. So among other things, I'm going to emphasize the different methodological perspective we have used so far to address this evolutionary process. So by definition, vector-borne diseases are diseases that are caused by pathogens that can be transmitted by mammals, insects, or other arthropods we collectively call vectors. So as you can see on this map, the range of these vectors and the disease they carry has been expanding over the last couple of decades. So vector-borne diseases are now a global issue, and there is no continent that's not affected by this type of disease. So in the case of the US in particular, ticks and mosquito-borne diseases have more than tripled in the United States since 2004. And you can see on this map, California fall within the hotspot of several of those vector-borne diseases. So it's a major public health issue across the state. So when we talk about mosquito-borne diseases, there are four key components. We have the environment that should be suitable for the development of mosquitoes and the pathogens. We also think about the pathogens that should be virulent enough to cause the disease, and should also have the potential to escape the immune system of the mosquito. But the last component is the mosquitoes that should have the vectorial capacity to carry the pathogens and transmit it to humans. So when we talk about this type of disease, we think typically about diseases just such as malaria, well-nice fever, for which we still do not have an efficient vaccine. And for some of those diseases, even the cure is not terrible. So in such context, the best thing to do is to target the mosquito vector. So there have been a diversity of protocols and techniques that have been designed to achieve these goals. For instance, the protocol we call integrated mosquito management is based on the use of a combination of methods to control mosquitoes population at the individual, household, or community level. And as you can see on this slide, I've listed several of those methods, such as the use of repellents, bed nets, source reduction of larvicides, biological control. So recently, another class of methods have emerged. So these methods are based on the release of modified mosquitoes. So these mosquitoes are modified to carry a factor that can either reduce the potential of wild populations to transmit the pathogens, or simply reduce the number of individual mosquitoes we have in nature. So, and there are four classes of methods that currently exist or are still under development. We have, for instance, the use of Wolbachia-infected mosquitoes, the use of female-specific lethal genes, the use of sterile insect technique, or gene drive mechanism. So all these methods have been widely deployed recently as a result of the expansion of the range of several mosquito vectors. So one of the current challenge is that we all know that mosquito won't just sit and see what's happening. They are going to react and try to adapt to all these control methods that are widely deployed. So one of the main objectives for vector control right now is to try to understand what's the underlying mechanism of the adaptation of mosquitoes to this wide range of techniques, and to precisely disentangle the role of different human impacts that can be intentional or not on the evolution of vector population. So when we think of disease-carrying mosquitoes in the US, there are two main groups of mosquitoes that are responsible for vector-borne disease in the US. We have populations that belong to the genus Culex, and that can carry diseases such as Weld Nile and several viral encephalitis. So on this map, I've shown the distribution range of three of those species: Culex pipiens, Culex kenkifelcetus, and Culex tarsalis. So we also have Aedes species, notably two invasive mosquitoes we call Aedes aegypti and Aedes albopictus, that can transmit diseases such as Zika, yellow fever, chikungunya, and dengue. So in all of those mosquitoes, there is some component associated with the role of human in their distribution and their vector stages. For instance, if we take the case of Aedes, we all know that increased international transportation has been an important factor in the spread of these mosquitoes around the world. So there's really a strong case to try to understand how humans contribute to, let's say, domesticate those species and make them an excellent vector of diseases. But right now we have an even more interesting model that can be used to understand the interplay between human interventions and the evolution of mosquitoes. So this model is that of African malaria vectors. Notably, as you can see on this slide, several species that occur in sub-Saharan Africa and that transmit Plasmodium pathogens in this region. So there are several reasons why those species are excellent model for studying the interplay between human impacts and mosquito evolution. So one factor is that in sub-Saharan Africa over the last few decades, there has been rapid changes in land use and land cover. Aspects such as urbanization, intensive agriculture, and deforestation has been expanded rapidly. And this type of rapid environmental change affect the habitats of mosquitoes, and they are challenged to a very strong factors associated with human intervention in the ecosystem. So the other aspect that makes these mosquitoes ideal system for studying the interplay between human and mosquito evolution is that in sub-Saharan Africa over the last few decades, there has been a widespread use of insecticides in large-scale malaria control programs. So the mainstay of global efforts to control malaria depend on two factors. One is the use of insecticide-treated net, and the other is the use of indoor residual spraying. So both methods are contributing to the spread of insecticide resistance among the population. As you can see on this map, red dots represent population that are resistant to several pyrethroid insecticides. And you can easily deduce that insecticide resistance occur almost everywhere throughout sub-Saharan Africa. So the last factor why we believe that this system is even more interesting for studying the interplay between human and mosquitoes is that we have significant resources for those species. A species like Anopheles gambiae, you see at the center of this slide, has become very attractive for many reasons. It's first of all an interesting model in terms of public health due to the disease they carry, but they also a model for people interested in other more fundamental aspects such as speciation. So these have been a factor that has driven a lot of studies based on these mosquitoes. So right now we have significant resources that have already been produced for those species. So the other important fact is that we have reference genome sequences for most of those populations. And the other interesting aspect, those mosquitoes also have a relatively small genome size, which is very important when you want to conduct, to perform experiments such as genome scan. And the last thing is that those mosquitoes are very flexible on an adaptive standpoint. And this adaptive flexibility fueled the rapid evolution. So to talk a little bit more about factors that account for this adaptive flexibility. So we have several types of polymorphism that are extremely abundant in those species. So recent deep sequencing of natural population show that Anopheles, there are more than 50 million SNPs in Anopheles gambia. In those species, we also have well-characterized patterns of chromosomal inversions. Chromosomal inversions are portion of the chromosome that are inverted as shown on the figure on the left. And those type of polymorphism have been well studied in the dipteran species. And they are involved in several type of process, including the adaptation to different environment. As you can see on this picture on my right, there is just a gradient of aridity in this country, and the distribution of one inversion we call 2LA, which is frequent in Anopheles gambia, goes from 0 in the forest to 100% in the arid savanna, which shows that this inversion plays a significant role in environmental adaptation in this species. So we also have another polymorphism called copy number variations. So portion of the genomes are duplicated, and recent study shows that this duplication is very frequent for genes, for instance, involved in pesticide resistance. So all this type of polymorphism represent a significant source of variation on which natural selection can act to adapt population to different contexts. So for instance, right now, Anopheles mosquitoes occupy an extremely broad range of larval habitat due to this type of adaptive potential. So if you look at the different type of habitat we can have, it goes from lakes, tires, rivers, even cattle footprints. So there are virtually no standing water that Anopheles mosquitoes cannot exploit in sub-Saharan Africa. So you have said that this is an excellent model, but it doesn't mean it's necessarily an easy model. We also have some technical difficulty. So the first challenge is that the most important malaria vectors in sub-Saharan Africa belong to cryptic species complex. So which means that we do not have obvious divergent phenotype between sibling species. So, and even when we try to study the phenotypic divergence in laboratory conditions, you are limited by the fact that there are no laboratory colonies for many species. So a species like Anopheles cambi at the center of the picture is relatively easy to colonize. But if you take other species, they have been very, very challenging to maintain in laboratory settings. So even if you succeed in having those lab colonies, you also go through another factor, is that the high level of genetic diversity we have in those species is an inconvenience for techniques such as genetic mapping. Typically, we think of genome-wide association studies or QTL mapping, which are really interesting tools to analyze genomes of mosquitoes and look for the relationship between phenotypes and genotypes. So in the face of this challenge, we have to think of a multidisciplinary approach to try to address the mosquito adaptation. So when I say we, I will first mention that this was a collegial effort involving several teams based in the University of Notre Dame, the University of Riverside, obviously, the University of Yaoundé, and other teams from Europe and the United States. So my colleague and I have so far think of 3 approaches to try to address this adaptive process. So the first approach we used was multi-scale ecological modeling and geostatistical analysis. So we also tested experimental evolution and transcriptome profiling, and we ended by sequencing a lot of populations to better understand the basis of the genetic basis of adaptation in those populations. So before I try to walk you through the different results we have obtained so far, I'll first mention one word on the geographic context of most of those studies. So the studies I will be presenting are based in Cameroon. So we have chosen this country because it's also nicknamed Africa in miniature due to its great environmental diversity. So if you cross the country from the south to the north, you will just come across the range of environmental diversity you can see throughout sub-Saharan Africa. So this gives a strong advantage when you want to study the biodiversity of mosquitoes. So the other advantage is that this environmental diversity gives rise to a very significant number of malaria vectors, as you can see on the map on my right. So this map comes from a paper published back in 2006, which shows that more than 20 Anopheles species are able to carry malaria parasites in sub-Saharan Africa, albeit at different vectorial capacity. So in my talk, I will be presenting data based on 3 of those species, which are really the most significant. Because recently, as you can see on my left, we have published some important data on other species that are still significant, but sometimes geographically restricted. So I'll go ahead and show the first experiment we did. So we are interested in understanding what are the environmental factors associated with the distribution of the 3 main malaria vectors that occur in Cameroon, which are Anopheles gambiae, Anopheles colosea, and Anopheles arabiensis. So the 3 species are morphologically identical species belonging to a species complex called the Anopheles gambiae complex. So to do this, we draw a transect starting from the dry savanna in the north all the way to the deep forest, and we sample more than 300 locations throughout this north to south transect using a technique called spray catches. Spray catches consist just in using pyrethroid to kill mosquitoes that are resting indoors and collecting them on white bed sheet, as you can see on this picture. So we also use geographic information systems to extract 16 eco-geographical variables that represented land use, land cover, hydrographic network, and human settlement. And we use a statistical approach called ecological niche factor analysis to assess the relationship between the occurrence of these 3 species and the 60 eco-geographical variables we have extracted. So these 3 maps show the habitat suitability profile of these 3 main species. So on my left, we have the habitat suitability map of Anopheles gambiae. So we have classified habitat suitability from unsuitable in white to optimal in red. So as you can see on this map, Anopheles gambiae looks to be a more generalist species, which is distributed throughout the country. And in the middle, we have the habitat suitability map of Anopheles colosea, which looks to be confined or to predominate in the southern part of the country, dominated by the deep forest. And the habitat suitability map of Anopheles arabiensis is simply the opposite. So they tend to prevail in the northern part of the Cameroon, which is dominated by the arid savanna. So you can see that I've highlighted one region with a red box at the middle of the map. So this region is a region that is densely urbanized in Cameroon. It mainly it's where you can is the location of the capital city, Yaoundé. So we saw that habitat suitability of the two species that diverged recently, Anopheles gambia and Anopheles colusi, overlap in those regions. And we were interested in using this opportunity to test a very long-standing hypothesis about the role of urbanization in ecological diversion of those species. So to do so, we use a sampling grid to extract 100 villages in and around Yaoundé. So the base map you see here shows Yaoundé in pink. It's a Landsat image which shows Yaoundé in the middle of the map, and the other shades of pink are just cloud contamination that occur when you are trying to get satellite image in these very cloudy regions. So the red dots are the different locations we sample in the 100 villages that surrounded Yaoundé. So the results show that there is a very strong segregation between the habitat of these seedling species. Anopheles colusia is predominant in the urban area, while Anopheles gambia tend to escape the urban region and occur exclusively in a rural area, as you can see with the green dots on the map. So to learn more about this segregation, we also draw a transect that crosses the city, starting from the first villages in the rural area all the way to the center of the city. And we sampled 16 villages across this urban to rural transect. So the result confirmed the stark segregation we have seen between the habitats of these seedling species. So these numbers represent the 16 villages we have sampled, and you can see that the frequency of Anopheles colusia, the species that adapted to the urban regions, goes from 0 in the rural area all the way to 100% in the middle of the city. So we got these results. These results were based on just, as I said, a small region around Yaoundé. So we wanted to know to what extent this can be generalized to the whole Cameroon or the whole southern forest regions. So to do so, we went back in our database and we extract several satellite images that we classify to try to collect a variable we call build environment. This variable represents a proxy for anthropogenic habitat disturbance. As you can see on this picture, build environment typically represents features such as bare soil, as opposed to more primeval landscapes such as the deep forest you see on the right of side of the map. So with this variable, we also go back in our database and we extract 300 sampling locations in South Cameroon, and we use a logistic regression model to study the relationship between the probability of occurrence of these two species and the build environment index. And you can see the result is very striking and confirm the relation that has been established based on the study around Yaoundé. There is a clear relationship between an anthropogenic factor such as the green environment and the probability of occurrence of both species. So we got these data at the moment where there was people were starting to deploy insecticide treated net at a very large scale throughout sub-Saharan Africa. So the first results that were coming up were showing that in urban areas especially, insecticide resistance tend to be extremely high. So one hypothesis was that perhaps in such region, there is a contributing factor to this increased resistance due to the fact that larvae that occur in this region are exposed to high levels of environmental pollutants. So this picture shows an example in Yaoundé, where you can see the typical breeding cycles really heavily load in other type of organic pollutants. So this type of hypothesis could be tested only by studying the genetic basis of this adaptation. So we do so. We start by conducting an experiment we call a reciprocal transplant experiment, and we use global transcriptome profiling to study the outcome of this experiment. So I'll try to explain what we did. So on the map on my left, we have selected 5 locations in Yaoundé, the urban regions, and 5 other locations in the rural area. And on my right, you see 2 typical breeding sites coming from these 2 ecosystems. And we have collected larvae to 4 instance and second instance that we have transplanted to different breeding sites. So on this table shows the type of experiment we did. For instance, 24-hour auto-translocations and 24 hours allo-translocations. And then we use transcriptome profiling to see the changes in pattern of gene expression due to this type of exposure. So the results show that we have typically several classes of genes that are overexpressed in populations that are adapted to urban area. But one interesting aspect is that genes that potentially function in stress response in Anopheles colony, including proteins of the glutathione transferase family involved in metabolic resistance to insecticides, were overexpressed in these populations, as compared to rural population that didn't have this type of changes in the pattern of gene expression. So we decided to go ahead and try to use another method to broaden our view about the potential genes that are involved in this adaptive process. So to do so, we use population group sequencing. So we went back to the field and we collect population throughout Cameroon in 30 something sites. And we use rat sequencing to identify around 10,000 SNPs that we use to scan the genome of these populations. So to make sure that the population we have samples represent the diversity within a species, we use several sampling techniques, such as the collection of larvae. We also sample active female at night and female that are resting indoor using spray catches. So when we scan the genome of this population, so first I will try to recall the principle of genome scan. So in principle, regions of the genomes that are affected by natural selection typically display low genetic diversity compared to the rest of the genome, and sometimes can also be strongly differentiated between populations compared to the rest of the genome. So we use this principle to scan the genome and identify regions that are outlier and that are thus candidates of targets of natural selection due to human intervention. So when you look at this map, we have plotted several types of summary statistics that assess the genetic diversity and the genetic differentiation in a population we have collected from Cameroon across 5 chromosome arms in Anopheles. So the take-home message of this plot is that if you look from my left to my right, we have highlighted several genome regions with a gray box. So these are the main selective sweeps we have in those populations. And the terrible thing is that all these selective sweeps contain factors that are associated with resistance to insecticide and pollutant. So the first selective sweep is associated with a gene called KDA, which is responsible for knockdown resistance to pyrethroids and DDT. The second is the ADL locus, which is associated with resistance to DL3. And we also have at least 2 clusters containing proteins of the glutathione transferase and cytochrome P450 genes that are involved in the detoxification of insecticides and other pollutants. So we did this test, as I said, using a rat sequencing to be able to analyze a large number of individuals. But we also select a subset of individuals that we sequence using the whole genome sequencing. And we got the exact same pattern. So we are very confident about the result we have obtained so far. So importantly, recently a consortium called the Anopheles GMB 1000 Genome Consortium, based in England, has also conducted a very extensive studies. They sequenced more than 10 populations from across sub-Saharan Africa. And when they scanned the genome of those populations, they obtained the exact same pattern we have observed in Cameroon. As you can see here, they have highlighted several selective suites that correspond to the KDR gene, the gene responsible for DL3 resistance, and several clusters of detoxification enzyme. So one other aspect is that what we know right now is that there are, at each selective sweep, you have several types of haplotypes that are distributed throughout sub-Saharan Africa. And the main question is to be able to understand what are the environmental drivers that affect the frequency of the different haplotypes we have at each selective sweep. So to give you an idea about how this selective sweep are structured, I will take the example of the selective sweep we have identified on the 3R chromosome, which contain a cluster of glutathione transferase enzyme and cytochrome P450. Here we have shown 3 locations in the forest region of Cameroon: the capital city Yaoundé, Douala, and the coastal region. So on my right, you see the distribution of genetic diversity across these 3 sites. And you can see that the drop of genetic diversity is very intense in Yaoundé. It is relatively significant in Douala, and it is almost inexistent in Yaoundé. So this shows that there is a very strong geographic structure across this selective sweep. And we really need to understand what's driving this type of structure to be able to understand how they contribute to the epidemiology of malaria in sub-Saharan Africa. So another consequence of this strong structure in haplotype is that they also affect the genetic differentiation within populations. If you go back and take the population from the same 3 locations, and instead of focusing on the selective sweep, we try to look at the whole genome, and we use around 10,000 SNPs to see how population will cluster. You realize that they cluster exactly the same way as what we have obtained within the selective sweep. So which shows that human-induced selection is also contributing to disrupt population structure within species, and this can have very strong consequences in terms of malaria transmission. So even within a city, I take the example of Yaoundé. These are 4 locations we have sampled in Yaoundé. And if we take the exact 10,000 SNPs and try to project them along a 3 PCA axis, and you can see as you can see on this graph, you see that the different individual cluster based on their geographic location within the city. So it shows that even within the same city, we still have a strong segregation among population. So for a species that occur typically in rural area like Anopheles gambiae, even in such context, you have a strong impact of human activity in their genetic structure. So in this picture, you see a gentleman she's doing gardening. This is a location around Yaoundé, known for intense gardening, where people used to use several classes of pesticides. during this activity. And we have realized that when we have attempted to study the genetic clustering within this species, we have seen a small subgroup corresponding to this geographic location, which is also emerging. This also shows that even urban agriculture is a contributing factor to disrupt the genetic structure within the species. So, based on all this data, what are the lessons we can learn from recent large-scale malaria control in sub-Saharan Africa? So we have seen that key entomological parameters, such as the distribution of species, their genetic structure, their resistance to pesticides, display fine-scale spatial and temporal structure. So we have also seen that human impact exacerbate the patchiness of vector, the parasite, and can also affect the geographic distribution of epidemiological parameters. So we believe that this should be carefully addressed in order to improve current methods that are implemented to reduce the impact of vector-borne diseases. So how we are thinking of this improvement? So we think that right now we really need to go for unifying protocols that integrate aspects such as landscape ecology, meta-population dynamics, and genomics to further understand the adaptation of mosquito population to different human interventions. So on this graph, I've tried to define what we mean by landscape ecology and meta-population dynamics. Landscape ecology just try to understand the spatial arrangement of species and their relation with their physical environment. On this map, we have dots that represent the distribution of a given species. So we just want to know if this distribution is regular, patchy, or random. Meta-population dynamics seeks to understand the relationship and connectivity between different patches. So these different figures show, for example, different patterns of exchange of population, different cluster of population, and that can be used to test different type of hypotheses in terms of how population within species exchange individuals and matter. So these two techniques work well in a geographic information system platform and have been used in the landscape ecology for several decades. So we think that if we combine these two approaches with genomics and other laboratory experiments, this can be a very powerful approach to further understand the geographic structure of mosquitoes and how human impact affect this geographic structure and their adaptation to the control methods that are currently deployed. So we have tried to take a concrete case to try to illustrate our point. So this map shows the distribution of Aedes type, which is a major vector for diseases such as Zika, yellow fever, chikungunya, and dengue. So on the map, we show the distribution of the abundance of this species in the United States, which goes from low in yellow to high in red. And you can see that there is a very strong cluster in Florida, for instance. So right now, some studies have been trying to understand how, since the species is not native to America, they are really interested in knowing how this colonization is moving. And we really need to see which type of population is contributing the most to the spread of this species across the United States. For example, on this map, populations that occur in New York have been hypothesized to originate from Florida. And this hypothesis has been based on population genetic analysis using microsatellite data. So we believe that unless we add other techniques such as landscape ecology, meta-population dynamics, and genomics, we cannot really come up with a very strong hypothesis about this pattern of colonization. So we believe that if we combine these three approaches, we can be able to, for instance, have more confidence in the detection of insecticide resistance. We can study the relationship between insecticide resistance and fitness. We can analyze genetic diversity and recombination rates among these species, and we can also understand the competitiveness of mosquitoes that are currently released throughout the United States to combat these mosquitoes. We really want to understand how competitive are they compared with the natural populations. So this is the type of multi-scale, multidisciplinary approach we are trying to advocate right now. So I'll end by showing one concrete case where we have used landscape ecology in a very in an operational setting. So as you know, in Africa, there are two main methods to combat malaria. As I say in the beginning, we have indoor residual spraying and universal coverage with bed nets. Indoor residual spraying use insecticides like carbamide and organophosphate that are more toxic and can be very costly. So you really need to know the different clusters of mosquitoes to be able to implement this type of method efficiently. So on this map, I show an example of the distribution of Anopheles DMB mosquitoes around Yaoundé. So we have done this sampling and we have implemented several geostatistical analyses to be able to delimitate the different clusters of mosquito abundance, as you can see on the map on my right. So the red contour plot represents a cluster of high abundance that can be targeted by this type of techniques such as indoor residual spraying. And this large cluster in blue in the middle represents a cluster of low abundance. So with this technique, we wanted to identify pilot regions for the implementation of this type of technique of malaria control. So this is a clear case that shows that even in terms of in a very direct operational setting, you can still efficiently use techniques such as landscape ecology. So I'll conclude by saying that we have seen that human impact are a major driving force of mosquito evolution. So in the beginning, I said that the interplay between human impact and mosquitoes affect virtually all types of disease-carrying mosquitoes. But the case of Anopheles species is even more emblematic. So we also highlight the importance of multidisciplinary approaches to address the fine-scale ecological and genomic changes in those mosquitoes. And we believe that there is really a necessity to improve vector control measures with tools that will address this fine-scale temporal and spatial dynamics. So I'll end by acknowledging several people that are listed here and several institutions such as the University of Notre Dame, the University of Yaoundé in Cameroon, UCR, and I won't forget a very interesting team I had in Cameroon, as you can see on this map. So with this, thank you for your attention, and I will be happy to take any questions.
Any questions for Colince?
Yeah.
If you wiped Anopheles with a city, say sterile technique, would DMB then colonize that city?
You mean if we wipe out Anopheles? Yes.
So sterile male just makes
it's a very tough question. So as we used to say, we don't know if it will remain an empty niche or if it will be invaded by another species. But what we know is that adaptation to urban area is very complicated, even for highly anthropophilic mosquitoes. So we don't really know how we can predict this in terms of let's say, across a time scale. But it will probably take some time for a species to get all the adaptive potential in terms of ecology and genome to be able to occupy this. So that's what we say. Yeah.
Have you thought about, or is there a way to spot that having like climate resistance?
Yes, yeah, we know that.
Colin,
Yeah.
in Davis, we can't hear the questions. Would you mind repeating the question?
Ah, okay, sorry. So she was asked, the first question was to know if we wipe out Anopheles colosea from the urban era. So is Anopheles gambiae going to invade this region or not? So that was the first question. And the second question is try to know if we can add other variables such as climate change to the data we have right now. Is it to improve the model or just to make it more? Yes, so adding climate change will make our data even more noisy. So you know that these are multi-dimensional data. It's very difficult to know which variable is really contributing the most, which variable having the most, the strongest contribution to your model. So that's why we focused on a variable that had, let's say, to us a much direct epidemiological impact, such as those associated with human interventions.
Any questions from Davis?
Do we know anything about the different vectoral capacities of the different members of the Anopheles species complex? Is Arabiensis better at transmitting malaria than Colince? Something like that?
Yeah, there have been very interesting studies conducted on this aspect for, did you hear the question? Okay, there have been interesting studies about this aspect. And as you know, vectoral capacity is a composite variable. And if you try to compare, for instance, Anopheles arabiensis and Anopheles colince, in terms of geographic distribution, Anopheles arabiensis has a broad distribution range. But if you take anthropophily, Anopheles colince feed exclusively on human hosts, while Anopheles arabiensis is much more generalist. So at the end of the day, it's very tricky to know who is really the best vector among those species. But usually specialists, vector biologists used to say that Anopheles gambiae is the first, and the next is Anopheles colince, and Anopheles arabiensis comes third, based on the combination of several factors, including the geographic distribution, the anthropophily, the sporozoite rate, and many other factors.
Colince, so yeah. I think this is referring to the 2014 Cassone paper.